And we saw there that, atoms form molecules, molecules form cells, cells give rise to tissues, tissues give rise to organ systems, and organ systems give rise to a whole organism, right? So we did atoms and molecules. Now we're going to look at cells and then how they form tissues. So cells of a similar type, having the same function, form a particular kind of tissue. So let's look at the 4 basic tissues that we have in the body. These are the 4 basic tissues, and they are formed by these 4 basic cell types. So we have epithelial cells, which will form epithelial tissue, connective tissue cells, which form connective tissue, muscle cells, which form muscle tissue, and nervous cells, which form nerve tissue, okay, nervous tissue. When a fetus is developing, there are 3 layers which are present in the fetus. So if I showed you a little fetus developing, the outermost layer, which is present here on the outside, this outermost layer is known as ectoderm. Ectoderm, yeah. E-C-T-O-D-E-R-M, ectoderm. There is a middle layer which is present, and I'm just going to kind of draw it, though it's not exactly anatomically correct, but I'm just going to show you a middle layer which is present. That middle layer is known as mesoderm. And inside, imagine this is the lining of your gastrointestinal tract, if I kind of show you like this. So like that, inside the body, the cavities, the lining over here is a 3rd layer which is known as endoderm. So these are the 3 basic, what we call germ layers, which give rise to all the tissues in your body. Ectoderm, mesoderm, and endoderm. Epithelial tissue arises from all germ layers. So the outside, for example, the epidermis of the skin arises from ectoderm. In some parts, we'll see lining some structures, it arises from mesoderm. And the lining of organs with cavities, it's endoderm. So epithelial tissue arises from all germ layers. Connective tissue arises from this middle layer, which is called mesoderm, as does muscle tissue. Nervous tissue arises from this outer layer. This outer layer actually kind of goes in and forms the brain and spinal cord and the nerves. So nervous tissue arises from ectoderm. Now let's look at each tissue. They are called germ layers, just like stem cells. These are called germ layers. In other words, they give rise to everything in your body. Okay? So let's look at epithelial tissue, the first type of tissue. Some general features of epithelial tissue. The cells are very closely packed and they tend to form continuous sheets, because epithelial tissue forms the lining of the outer surface of your body or the lining of the inner cavities in your body. For example, the lining of the gastrointestinal tract, the lining of the respiratory tract, the lining of the reproductive or urinary tract. So the lining inside, or on the outside of your body, for example, the epidermis of the skin. So you have a continuous sheet of cells and they are very closely packed. So imagine if I looked from the top, the cells are closely packed like this. Very closely packed, forming a sheet. And this sheet has to rest on something. So all these cells sit on a foundation which is known as the basement membrane, which is made up of connective tissue. So basement membrane is made up of connective tissue and these epithelial cells sit on top of that. So if I was to look at it from the side, imagine if I took a section from here and I looked at it from the side, imagine this would be the basement membrane and these are the cells sitting on top. with their nuclei. So you can see that they have an attached end. This end is resting on the basement membrane. This top part is called the apical surface. So it's the apical or upper surface. This is free. So imagine in your skin, for example, so if I was to draw a person, oh, that's really horrible. Okay, so let's take the skin. The skin has many layers, but just for now, let's imagine it had one, it has this connective tissue which is called the dermis, and here's the epidermis. So imagine this is the basement membrane. Here are the cells of the epidermis, which is also called epithelium. So you can see that one part is resting on the basement membrane, one surface is free. So epithelium, remember I said, lines the outer surface of your body, or it lines the inner organs with a cavity. So imagine, let's say that I draw, here's your mouth, and here's the gastrointestinal tract, right? It's like a tube. So if I take this tube out and I show you here, can you imagine this is the basement membrane, here are the cells. So they have one surface which is free, which is towards the cavity of that organ, right? So imagine you have a tube like this. So the one surface of the cell will be on the basement membrane, the other will kind of face the cavity. So that will be a free surface. So they always have an apical free surface and one attached surface which sits on the basement membrane. Another important fact, epithelium does not have any blood vessels. It's avascular. So how does it get its nutrition then? It gets its nutrition from below, from the connective tissue below. So it's sitting on the basement membrane, and below it are other tissues like connective tissue, muscle tissue. So it gets its nutrition from below through diffusion. So you can understand that if there is epithelium which is just single layer like this, it can get nutrition from below because this is all connective tissue. Can you see when epithelium becomes many layered, the top layer of cells are very far from the nutrition. Can you see that they will not get adequate nutrition, so they'll begin to die, which is exactly what happens. So when you have very, very thick epithelium with many layers, the top layer of cells actually die. And we'll see that that's what in your skin, for example, peels off as keratin. That little, when you scratch your skin, you get the little flakes, that's keratin. And that's because of these dead cells which are present, because the epithelium is so thick that the top layer is very far away from nutrition, so it dies. Okay? However, while it doesn't have a blood supply, it has a very good nerve supply. And because epithelium is constantly abraded, see, imagine your skin is constantly abraded inside your organs, the lining of those hollow organs like your, say, mouth, your stomach, your esophagus. You eat food of different texture, different temperature. So that can kind of, when you drink something really hot, you know how your mouth gets scalded. When you eat something which is kind of little sharp, it might scratch your oral cavity. So because of that, epithelium tends to regenerate very, very fast. It has very good regenerative properties. So it has rapid cell division. It forms, as I said, the lining of your outer surface of your body, the epidermis of the skin, or the lining of hollow organs inside. Epithelium also forms something known as glandular epithelium. It forms glands. So imagine if you took, let's say you took the small intestine. So if you look at the small intestine, it looks like this. Okay? And let's say I took a small part of the intestine and I kind of just enlarged it. So if this is the cavity, so this is what is known as lumen. The lumen means the cavity. So here is the lining, and here are the epithelial cells. Then underneath it will be connective tissue. I'm only drawing a part. And then there's muscle below it. And then there's some more connective tissue. So this is the wall of the small intestine. Now in certain parts, if I just enlarge this, in certain parts of the body, if this is the epithelium, what happens is it dips down inside and comes back up. So where it dips down inside, here it forms glands. So here what it would do is it would dip down inside and come back up. It would dip down inside and come back. And here is where it forms glands. What do glands do? They secrete, right? So secretion will be poured into the lumen. From here it will get into the lumen. So glands are formed from epithelial tissue, from the epithelium. Some functions of the epithelium. So you tell me, what can epithelium do? What are some of the functions you think it is capable of? Okay, very good. Secretion. So the cells are facing the lumen. They form glands, so they could secrete. Good. Then? No, they are simple cells, so they won't be able to move fluid much. They can't, they're, cells very close to each other. Cilia is needed to move that part. What? Yes? Protection. Very good. Protection, because it is forming the lining of your outer surface of the body or the inner lining, so it is protective. So wherever epithelium is, where you need more protection, you want epithelium to be thicker, right? So where you don't need that much protection, epithelium will be thinner. So protection is one. What else do you think it's capable of doing? Yes, I mean that's not a function, that's just a property. Because it is lining, it secretes, it can also absorption, right? Like your gastrointestinal tract, it secretes enzymes, they break down foodstuff, and you can absorb the nutrients through the epithelium. That's the only way it can get into the bloodstream, isn't it? Through the inner lining. So it has absorption is another property of epithelium. Pardon? Reproduction, that's a property of the cell, not just limited to epithelium. So that's because it has a nucleus, it'll capable of reproduction. But these are some main properties. Since it forms glands, again secretion is done by that, and that secretion could be enzymes, different substances like mucus, which will trap foreign particles. So that all of that comes under secretion. So secretion, protection, absorption are some main properties of epithelium. Okay? Okay, so I actually gave this answer out, but let's answer. How do you think it derives its nutrition, even though it's avascular? So let me see if you were listening to me. Through the underlying connective tissue. Yes, that's right. Okay, again, this one. Why does it regenerate rapidly? Because it is constantly abraded. I told you, because it's constantly exposed to friction. The more specialized a cell gets, the less it regenerates. If it doesn't have a blood vessel, have blood vessels, actually regeneration is not so good. But because epithelium is very close to blood supply, it can get enough nutrition to regenerate. Not because it has many layers. Some epithelia have only single layer. It's because it's constantly abraded. Because of friction, it's removed. So therefore, you want to make sure that you don't have any area which is devoid of epithelium where you should be. So it's regenerated very, very rapidly. Yes? Yes, because when you have wounds, when they heal, especially in the skin, the wider the area between the two ends, the more difficult it is for cells to actually come across. So by putting that, it kind of, you're helping those cells travel across. Yeah, that's what they do. Now here, classification of epithelium. Wherever you have classification of anything, it's really important you know how that tissue or that whatever I'm talking about is classified. So here, epithelium is classified based on two criteria. One is how many layers are present in the epithelium, and second is by the shape of the surface cells. So by the shape of the surface cells and by arrangement of layers. So by arrangement of layers, if the epithelium has a single layer, it is called simple epithelium. If it has many layers, it's called stratified epithelium. The reason being the word strata means layer. So stratified means many layered. In between the two, you have something which is called pseudostratified. Pseudostratified means it's in between. It is not truly, pseudo means false, so false stratification. It's not truly stratified, but looks like it is. And when we describe it, you'll understand. Then based on the shape of the surface cells. So in simple epithelium, it's a single layer, so it's that layer based on the shape of that layer. But if it is many layered, whatever the top shape of the top layer of cells is, based on that we call it, we give the epithelium its name. So here we have three main types of epithelia. Squamous epithelium is where the cells are very thin, very, very thin. So if you look at it from the top, it kind of looks like a fried egg. You know how in the egg the yolk is a little bit raised and the white is flat? So if I take a section through an egg and I look at it from the side, can you understand this is what it's going to look like, right? And the nucleus is usually will be present here in the thickest portion. So this is what a squamous cell looks like. Squamous meaning pavement like, very thin. So this is squamous epithelium. So if you have squamous epithelium made of just one layer, we'll call it simple squamous epithelium. We use both classifications together. So we use this and this together. So simple squamous epithelium. If on the other hand we have an epithelium which has many layers, but the top layer has to be squamous, so it is thin like this, flattened out, here we will call it stratified squamous. Do you understand? Stratified squamous, meaning many layered and the top layer is squamous. Cuboidal epithelium is where the cells look like a cube. So if you look at it from the side, they are as tall as they are wide. The nucleus is usually rounded. Very little cytoplasm above the nucleus. Most of it is on the side. So they look like cubes. So again, you can have simple cuboidal, meaning it's a single layer of cuboidal cells. You can have stratified cuboidal, where the bottom layer could be anything. Usually it will be columnar or cuboidal. But the top layer has to be cuboidal. So that's why you call it stratified cuboidal. Columnar epithelium, where the cells are like columns. So here the cells are taller than they are broader. And the nucleus could be rounded or it could be rod shaped. But you'll always see plenty of cytoplasm above the nucleus because the cell is taller than it is broadened. So this is cytoplasm above the nucleus. Again, you can have simple columnar epithelium where it's a single layer of columnar cells. You can have stratified columnar epithelium where, many layers, but the top layer is columnar. this type is, while I've included here, I really should have put an asterisk. This is really not a shape of a cell. Transitional epithelium is a different type of epithelium. It's a stratified epithelium. And it's only seen in the urinary tract. And the cells are very different. So when we do transitional epithelium, I will describe it. So it's really not based on shape, but just that it is present in one area, and ideally this name should have been changed. It should have been called urothelium, so that it tells you that it is present only in the urinary tract. Okay, but it's not. It's still called transitional epithelium. It's not really a transition from simple to stratified, really. So I don't know why they got that name. But when we describe transitional epithelium, I'll talk about it. Yes. Which one? Yes. Oh, sorry.
It's okay. On the columnar, the very top part is the cytoplasm? Or is it
No, you have cytoplasm in columnar epithelium. You have cytoplasm everywhere. You have both on the sides, but you also see cytoplasm on the top. In cuboidal epithelium, you'll only see cytoplasm, mainly along the sides, very little on the top. Cytoplasm surrounds the whole nucleus. So you have cytoplasm everywhere, but I'm saying here you can see a lot more cytoplasm on the top than you can when you compare it to cuboidal epithelium. Okay. So actually, let's stop here and I'll start. So the first type of simple epithelium we are going to look at is simple squamous. So the cell is very flat, and squamous means like a pavement. So here's the cell that you're looking at, and I told you last time, it looks like a fried egg. So if you see it from the side, you can see that there's very little cytoplasm, and only where the nucleus is, that area is slightly raised. When we take a section of this kind of epithelium, we will see that it kind of rests on a basement membrane, and you can only see the nuclei of the cells. They are also flattened. You can barely make out the cytoplasm. Only if you went under very high power can you see the cytoplasm like you can see up here. So flattened cells with very little cytoplasm. Examples where you find this kind of epithelium is in the air sacs, the alveoli of the lungs, in the glomerulus of the kidney, which is here, where filtration takes place. In the blood vessels, this epithelium lines blood vessels, and it has a special name where it's lining the blood vessels. Instead of calling it epithelium, we call it endothelium. So lining of blood vessels is called endothelium. And where it lines even the synovial joints and the serous membranes. You all did the serous membranes. Remember the parietal, pleural, and pericardial cavities, which had a visceral and parietal layer. There also the name changes. There, lining the serous membranes, this is known as mesothelium. Okay, mesothelium. But everywhere else it's just called simple squamous epithelium. The name just changes in the blood vessel lining. Again, it's very typical. You can see just the nuclei. And in mesothelium also it would be the same. Now, where do you think you're going to find this kind of epithelium? Okay, well, half the class got it right. Think of it, it's very thin epithelium, very little cytoplasm, so it cannot secrete. In order to secrete, remember the cytoplasmic organelle, which is the organelle which packages and secretes, Golgi body, right? If your amount of cytoplasm itself is very little, you're not going to have too much Golgi body. It cannot secrete. protection is needed. Now, this is very thin epithelium. If you had to build your house, would you have thin walls or would you have thick walls? So you need, for protection, you need something to be really thick. So the epithelium which protects is usually stratified, many layered. Areas where diffusion occurs. Remember, diffusion requires substances to go from an area of high to low concentration through a semi-permeable membrane. If the membrane is too thick, it's going to take trouble to pass across. Imagine like you are pouring water. If you pour it through a cheesecloth, that's really good. But if you took tarp, which is really thick, it's going to be very difficult to pour water through. So this is a very thin epithelium. So it allows for things to pass through easily. Diffusion is one such area, which is why you see it in the kidney glomerulus, because blood gets filtered over there. You see it in the alveoli of lungs because gas exchange needs to take place, so gases diffuse through. You see it in the blood vessels because at capillary level, again, diffusion of gases occurs at the tissue part. And so that's why this is areas where you see need diffusion. So very, very thin epithelium, not where support is needed. Again, for support you need something a little thicker, not something as thin. So always try to kind of use common sense and relate function with what the structure is. Let's look at the next epithelium. Here the cells are cube-like, meaning as tall as they are broad. So that's why it's called cuboidal epithelium. So notice up here it again rests on a basement membrane. So you see the nucleus. So the cell is as broad as it is tall. So with the result, the cytoplasm is kind of present more on either side of the nucleus, very little often on the top of the nucleus. So above the nucleus you may rarely find here. This picture shows cytoplasm here also. But if you look quite often, like look at this picture here, notice there is cytoplasm on either side of the nucleus, but above the nucleus you see very little cytoplasm because there's no space over there. So these cells are as tall as they are broad, usually seen in small ducts, seen on the germinal epithelium of the ovary. You have a list of structures where it's seen in your notes, so I'm not going to go through that. So my suggestion to you is make flashcards, write down where the epithelium is found. This kind of epithelium, since it's a little thicker than squamous, will be seen where you need some support. You need to keep that area patent. Patent means open. That's why it's seen in ducts. Ducts are like tubes. You want to keep the tube open. You don't want the tube to collapse. So you don't have a very thin epithelium, but you have something with a little bit more structure to it. So cuboidal epithelium is seen there. Compared to this, the next type of epithelium, as the name suggests, is called columnar. That means the cells are like columns. So they are tall. So they are taller than they are broader, as you can see up here. So a lot more cytoplasm will be seen. So you see cytoplasm on either side. You see cytoplasm above the nuclei as well. And the nuclei may either be rounded like this or they may be rod-shaped, as you can see up here. They're rod-shaped here. Very often this kind of epithelium has in between the regular columnar cells. You might see cells which look empty like this. Can you see these? They look like little empty cells. Such cells are really single-celled glands and known as goblet cells. So these are single-celled glands called goblet cells, and they secrete mucus. When we process the tissue, we use alcohol, which tends to remove this mucus, which is why these cells look empty or frothy. Now, when we did the cell, you remember in one of the features of the cell was the cell had microvilli. A cell could have the plasma membrane. One of the adaptations of the plasma membrane was the plasma membrane could have microvilli. And the function of microvilli was to increase surface area. That's correct, right. So columnar epithelium often has microvilli because again it's seen in areas, since there's so much cytoplasm, it's seen in areas where secretion is done because it has enough cytoplasm to send stuff out. It's also seen in areas where absorption is required. So obviously if you increase surface area, you can increase absorption. So here you can see these microvilli which are present up here. The arrow is pointing here to microvilli. And what a microvilli on a cell will look like is, if you take a cell like this, the microvillus is small, little invaginations where the membrane is folded. Now, when you look at it under the light microscope, you won't see microvilli so clearly. What you will see is the cell kind of looks at the top. It tends to kind of give a little appearance, a fuzzy appearance, as you can see here. Look at it, it's kind of looking fuzzy. This kind of fuzzy appearance is known as brush border. So microvilli give rise to the brush border appearance in cells. So they further increase the surface area for both secretion and absorption. So the gastrointestinal tract is one area where you see columnar cells with microvilli. Very often you'll see goblet cells as well, because in the gastrointestinal tract the mucus helps to neutralize acid from the stomach. Other areas, look in your notes to see where they are found. Now, not all columnar cells have microvilli. Some may be very just simple columnar, like you see here, with no microvilli. And some columnar cells don't have microvilli, but instead they have cilia. So you remember that was another adaptation of the cell or plasma membrane. So if you look at microvilli, this is what microvilli look like. If you look at cilia, here is the cell, the cilia are on top. So look at that, they look very different. So can you see that? So this is cilia lying on top, and some of you in the lab today saw the cilia. So cilia is present on top, thin hair-like projections from the cell. So they're very thin, they don't increase surface area or anything, but they help to, actually what they do is they beat in a particular direction, and they help to move particles around or move fluid around. So they set up fluid currents and help to get rid of substances that the body does not need. So ciliated epithelium is seen in the respiratory tract. So you tend to see cilia in the respiratory tract. The epithelium is not simple columnar. It's pseudostratified there. But one good example is the fallopian tube, the uterine tube in females. They have simple columnar ciliated epithelium. That helps to push fluid, and that fluid sets up currents, and the idea is to get the fertilized egg going towards the uterus so it can implant and grow there. So simple columnar ciliated type of epithelium is seen in the fallopian tube. Just cilia, we'll see in the next slide, that's seen in the respiratory tract, and another kind of epithelium which also happens to have cilia. So you can have columnar epithelium, just simple plain columnar, or you can have columnar with cilia, or columnar epithelium with that brush border, which is microvilli. This type of epithelium is called pseudostratified, and remember when we were doing the way we classify epithelium, I told you pseudostratified means it's not true layering, it's just that the cells are at different levels. So with some are short, some are tall, so with the result the nuclei kind of tend to look like they are at different levels, giving a false sense of stratification or layering. So all cells reach the basement membrane because some are small, some are tall. So if you look up here, look at this cell. This is really tiny cell. Look at this. This is very tiny. Look at this cell, much larger cell. But notice both of them are touching the basement membrane. See here, small cell, much larger, touching the basement membrane. So all cells touch the basement membrane. So it's not true layering. So that's why we call it pseudostratified. Only the nuclei are at different heights because based on the size of the cell. Larger cells tend to have a nucleus a little bit higher. Now, very often pseudostratified columnar epithelium also has cilia, like you can see up here. So it may have cilia. And typically, often pseudostratified epithelium has goblet cells. So this kind of cells which you can see look empty. So then we would call it pseudostratified ciliated columnar epithelium. So then it would be known as pseudostratified ciliated columnar epithelium with goblet cells. Not always does it necessarily have to have cilia. But this kind of epithelium, pseudostratified ciliated columnar, is seen in the respiratory tract. So all of the respiratory tract, you can understand, will be lined with this kind of ciliated epithelium, because you want the cilia to remove anything that you breathe in or something that goes down. You want to cough it out. In some parts of the body, you may just have simple pseudostratified columnar, no cilia present. May or may not have goblet cells. But the most predominant area where we see pseudostratified epithelium is the respiratory tract. And there it has cilia and it has goblet cells. And you can understand why. Because these goblet cells produce mucus, which is very thick, and that will trap cell debris and prevent that debris from going into your larynx or voice box. So it'll kind of, you tend to cough it out. Now we come to layered epithelium, also known as stratified. Strata means layered. And the epithelium is called stratified because it's more than one layer. And the second part of the name of the epithelium is based on the top layer of cells. The bottom doesn't count. Whatever is the topmost layer of cells, then we name it that way. So here you can see stratified cuboidal. Look at the top layer of cells. They are cuboidal in nature. The bottom is also cuboidal, but it could be columnar. It doesn't matter. Usually it will be columnar or cuboidal. So usually this kind of epithelium is seen in two layers. So it's just two layers. Sometimes you may get a few more. And again, because it's a thicker epithelium, it tends to be seen lining larger ducts where you need to support it. So really large ducts in your sweat glands and in the glands or your exocrine glands, like salivary glands and so on. You will see these huge ducts with a double layer of cells, and the top layer is cuboidal in nature. Stratified columnar, I know this picture kind of gives you many layers of cells and the top layer being columnar. Usually this epithelium, when seen, usually tends to also, like the cuboidal, is made up of only two layers or maybe three maximum. The top layer, of course, is columnar, but it's not a very common epithelium. This kind of epithelium is rare. It tends to occur in patches. That means in certain areas it will be columnar, whereas the rest of the area it may or may not be columnar. It could be cuboidal. So it's kind of like a patchy epithelium, not seen as frequently. When seen, again, it's seen in large ducts of exocrine glands. So here you can see the top layer being shown as columnar. This epithelium, transitional epithelium, this epithelium is not named based on any type of cell shape. Ideally, this is kind of an epithelium all by itself. It's a stratified epithelium. Ideally, the name should have been what is called urothelium because it belongs to the urinary tract, and it's only seen in the urinary tract, in the ureters, the urinary bladder, and some part of the urethra. So it has many layers of cells, as you can see. And you can see that the bottom layer is closely packed, columnar to cuboidal cells. And then we have cells in between which are shaped like a pear. So a pear-shaped cell. So the cells look like this. And there are many spaces in between. Look at these spaces. So they leave many spaces in between. And there's a reason why you have these spaces. And then the topmost layer of cells, you can see these top layer of cells, they're very large cells. They're really plump and large cells. And they are known as umbrella cells. Because if you look at one cell, it kind of looks like a little umbrella, like this. Here's the nucleus. This kind of epithelium, as I said, is present in the urinary tract. And the idea of having these spaces is that in a relaxed state, the epithelium height is tall because these spaces can be seen. But let's say it's seen in the urinary bladder and the bladder fills. As the bladder fills, it stretches. So what happens is that this cell will drop into this space. This cell will drop into this space. This will drop into this space. This will come down here. So then these cells kind of drop into the spaces. So the epithelium is capable of stretching and its height becomes less, because that's why you have these spaces. So this is one epithelium which is capable of stretch to accommodate large volume of fluid. And another feature of this epithelium is that this top layer of cells, they secrete a little sort of substance which looks, if you can see it well in a slide, you can see it as pink. This substance that they secrete is called cuticle. It's impervious to water. That means it doesn't allow water to pass through. So this kind of protects the urinary tract from water which is present in the lumen of the tract entering into the walls. So we use the word, this cuticle makes this epithelium impervious to urine. Impervious means urine is mainly water with some salts in it, but it doesn't allow water to pass through. If it did, it would get into in between these spaces. So that's the function of the cuticle, to make the whole lining waterproof. And as I said, it's capable of stretch because of these spaces. The next type of stratified epithelium is called stratified squamous epithelium. It's multi-layered, many layers, and the top layer is very thin and flat, like squamous cells. So that's why it's called stratified squamous epithelium. Here there are two kinds of stratified squamous epithelium. One is called stratified squamous non-keratinized. The other one is called stratified squamous keratinized. See the difference between the two. In both of them you have many layers of cells. The top layer of cells is flat. In the non-keratinized type of epithelium, you can see the nuclei right up to the top layer of cells. So you see the nuclei right up to the top layer of cells. This is an example of stratified squamous non-keratinized. Look at the many layers. As we are going up, the cells are getting flatter and flatter, but right till the top, you can see the nuclei of the cells. The cells are still living. This kind of epithelium is also known as wet epithelium because the surface is kept moist. In the next variety, which is called stratified squamous keratinized, many layers of cells, the cells getting flatter and flatter. You see nuclei up to a point. Beyond that, you don't see any nuclei. If you look under the microscope, you may see cell margins. You may see if you're lucky, you may see cell margins like this, but you don't see nuclei in them. So it kind of gives a homogeneous pink appearance, because these cells, as they are going away from the basement membrane, and from here is where they get their nutrition in the connective tissue below, they're getting further and further away. So they lose their nutrition and die. So all of this then becomes one homogeneous substance on the top, which is called keratin, which is a protein. So this is all dead over here. This kind of epithelium is also known as dry epithelium because the keratin flakes off, and this is seen in the epidermis of the skin. Your skin has two parts, epidermis and dermis. The epidermis is where you see this. So all the skin flakes that fall on your mattress, that's all the keratinized cells which are shedding off. And here's a slide of that. So this part is, you can see many layers up here till this point. You can see nuclei. Beyond this, notice you can't see the nuclei. And here, this is just that this part is just torn when the slide was being made. It just got torn. See, notice up here, you don't see this pink homogeneous layer. You can see nuclei up to the top. Here you see nuclei just up to here. Beyond this, you don't see nuclei at all. So this is keratinized, seen in the epidermis of skin. This kind of epithelium, non-keratinized, you can understand it's wet because it's present in your oral cavity, esophagus, anal canal, vagina, all these areas where protection is needed. Because, you know, like you eat substances of different texture, different temperature, that can kind of damage your epithelium. So you need a thick protective epithelium. Skin also, and in skin you don't want it to be wet at the top. You want the cells, since they are so thick, they flake off, and from below then they keep getting replaced. So, what do you think would be the prime function of any stratified epithelium? And I gave you the answer actually. The prime function, the thicker the epithelium, obviously it would be protection. I just told you. Secretion and absorption would be seen in epithelia. In order to secrete, imagine if the epithelium is so thick, like you see here. To secrete, it has to travel through so many layers and then go to the outside. It makes it much easier if the epithelium was just a single layer, like columnar cells. So columnar epithelium is more secretive and absorptive, whereas thicker stratified epithelium is more for protection. Now, epithelium also sends in glands, and these glands are called exocrine glands because exocrine means they have a duct which allows them to pour their secretions into the lumen of the organ. So let's say, for example, you were taking any epithelium, say some simple columnar epithelium anywhere, like is shown here. So here's the epithelium, columnar epithelium, and this epithelium during development goes inside, makes a little sort of, this going inside, the word we use is invagination. And here it makes a little gland and a duct. This lower part is the glandular part, which means which secretes. This part is the duct, which just allows the secretions to pass through and come out. So you can see how the gland would be. It could be a very simple part of the gland. It could be like this. The gland could be this way, or the gland could, these are the various ways that any gland can be. And for in the lab today, some of you saw sweat glands, and they are basically like this. The only difference being a sweat gland kind of gets curled up more this way. So this is how it is. So when we take cut sections of glands, even if we take cut sections, we'll see glands like this. And most of the time, glands are typically lined by cuboidal to maximum columnar epithelium, usually cuboidal, so that they can secrete, and the lumen is not, the taller the lumen, the taller the cells, the smaller the lumen. You don't want that to happen. You don't want to block the lumen. So you want to kind of keep it open. So usually glands tend to have cuboidal cells so that they can pour their secretions and the lumen or cavity remains wide. So such glands which have a duct through which they pour their secretions are known as exocrine glands. Sometimes glands develop, they may develop from epithelium, but not necessarily. So sometimes when they develop from epithelium, they go down like this, and after some time what happens, this connection kind of goes off. So the epithelium remains on top and the gland remains below. So such glands which don't have a duct, so they don't pour their secretions into the cavity, are known as endocrine glands. So they are often also called ductless glands because they don't have any duct to pour their secretion. So where do they pour their secretions? What they do is these glands pour their secretions into blood vessels close by. So whatever they secrete, they pour, that goes into the blood vessel close to it, and then from there the bloodstream takes it far away. Most of these endocrine glands produce hormones, or all of these endocrine glands produce hormones, which we'll do when we do the endocrine system in ANP2. Now, the way the glands secrete, the way they pour their secretions, is in three types, three ways of pouring their secretion. Majority of glands pour their secretions by what is called a merocrine secretion. So here's a cell, which is a cell in the gland. So up here, any one of these cells, so any one of these cells if we take, it has all these cell organelles. So what it does is that, and most of the secretions are enzymes, or they may be hormones, like we said. So what they do is that the rough endoplasmic reticulum sort of makes up those substances, which are usually protein in nature, transfers it to the Golgi complex, which makes these little secretory vesicles, and they go, and by exocytosis the secretion is poured out. So this kind of secretion is known as merocrine secretion. So most glands use this method of pouring their secretions. The second type is called apocrine, apo for apical, where the apical part, so you still have these vesicles formed, but then the apical part kind of gets pinched off during the cell secretion. The apical part gets pinched off, and then the cell membrane comes back together, and then the cell continues its job. They used to think that apocrine glands were, in human beings, they thought that the glands which are present in our pubic region and the glands which are present in our axilla or armpit were apocrine kind of glands. Now they're not so sure about it. They still say that most of them are probably merocrine type of glands, but you may have a few apocrine glands. They're not as common in human beings as they are in other species. But this is another method of secretion. A third method of secretion is where the entire cell of that gland just ruptures and all the secretion comes out. So that cell dies, and below cell it kind of is replaced by mitotic figures in the lower cells. So a new cell is formed. That secretes and just ruptures and dies. New cells keep getting formed. Such a kind of gland is called a holocrine type of gland, and the type of secretion is called holocrine. In our body, sebaceous glands, which you'll do when we do the integumentary system, some of you did it in the lab today. So sebaceous glands are one example of holocrine secretion, where the entire cell in the sebaceous gland just ruptures, and then it's replaced by other cells which are at the bottom. So these are the three modes of secretion. The next type of tissue we are going to look at is connective tissue. So here's the difference between connective tissue and epithelium. Remember, epithelium was cells which were present close together. They were packed close together, so they were as sheets. And they rested on something which was called the basement membrane. And that basement membrane basically is connective tissue. So connective tissue is like a foundation on which the epithelium can rest. Apart from that, some features of connective tissue, they have to have their own cells. These cells are not necessarily closely packed with each other. They are distributed, but they are different types of cells. They have fibers which are like threads, and they have a substance in which the fibers and the cells are embedded. That substance is called the ground substance. That ground substance and the fibers are secreted by the cells. So fibers and ground substance together are called matrix. So if I give you an example, so imagine, okay, let's put it this way. You took some jello, okay? That's your ground substance. So that's your jello, that's your ground substance. And then in this, let's say you threw some Twizzler sticks. Okay. Those are your fibers. And then you took some M and M's and threw that inside as well. Okay, those are your cells. Now what you got is connective tissue. Got that? Okay. So I said jello, but it could be really very, very thin and fluid, like some sort of a custard pudding. That could be a ground substance, where it would be very liquid. It will be jelly-like if you have jello, or it could be real solid as well, like we'll see. The ground substance in bone, for example, is really, really hard and solid. So the consistency kind of varies from liquid. Liquid is like in your blood, for example. The plasma, which is the ground substance, is liquid. In cartilage, the ground substance is jelly-like. And in bone, as I gave you an example, the ground substance is solid. So that consistency varies. Connective tissue does not occur on a free surface. Remember, epithelium had one free surface. The apical surface was free. Connective tissue does not occur on a free surface. It occurs below the epithelium, or it occurs between muscle or below muscle, but there's no free surface to it. It has a good nerve and blood supply. Remember, epithelium didn't have a good blood supply, but this does, except cartilage and tendons, which tend to have a poor blood supply, so they don't heal as fast. So this is kind of a general diagrammatic picture of what connective tissue would look like. So here, this space, this kind of pale colored thing, this area is the ground substance. That's the jello. Okay. And then in this, you have these different fibers which are thrown in, the Twizzler sticks. They could be put in bundles, or they could be separate. When they kind of run in bundles, we call them collagen fibers. So collagen fibers tend to run in bundles. They are most plentiful. Then we may have single fibers present. These single fibers could be elastic fibers. So these yellow ones, as you can see here, these are elastic fibers which tend to branch. So if you look here, elastic fibers may tend to have a branch. Collagen fibers don't branch. The bundles may branch, but the individual fiber won't. Or you may have very thin kind of, you can see these purple, very thin type of fibers. These are known as reticular fibers. So this very thin purple color is reticular fibers. Reticular fibers are not as plentiful, usually form a framework, and are seen in lymphatic organs. Collagen fibers are seen in all connective tissue. In lymphatic organs, you'll see a lot more reticular fibers, maybe less collagen fibers. And you'll see elastic fibers in area where you need some elasticity. So we'll see, for example, when we have something called elastic cartilage, we'll see more elastic fibers then. In the connective tissue of very large blood vessels which need to stretch, again we see a lot more elastic fibers. So that was ground substance and fibers. Let's see the various cells that we see. The predominant type of cell that you see in connective tissue is called a fibroblast. That's the one which gives rise to the fibers and the ground substance. Fibroblast. It's kind of star-shaped. It's the predominant cell of connective tissue, fibroblast. Apart from fibroblast, you may see all of these other cells. Like, for example, you may see macrophages, which are phagocytic in nature. So macrophages are phagocytic. So they take care of any foreign body, foreign object that has come there. You may see fat cells in connective tissue. They are called adipocytes, and together they form their own type of connective tissue called adipose tissue. You may see some blood cells like eosinophils, plasma cells, neutrophils. You may see these mast cells. These mast cells are usually take part in allergies because inside they have granules which liberate a substance known as histamine, and they liberate heparin as well. The granules produce histamine and heparin, and so when these cells are stimulated, they release that substance. So mast cells liberate histamine and heparin. So you will in certain areas see, apart from fibroblasts, macrophages are found pretty predominantly in many areas. You'll see in fat tissue, you'll see a lot of fat cells. Then in areas you'll see eosinophils, plasma cells, neutrophils, and mast cells. So these are some of the cells that you see in connective tissue, but as I said, the predominant cell of connective tissue is fibroblast. All of connective tissue comes from a primitive mesodermal cell which is called mesenchymal cell. Remember we did those germ layers? So in the primitive germ layer there is a cell called the mesenchymal cell which gives rise to all the components of connective tissue. So let's look at some types of connective tissue. Embryonic connective tissue, obviously it's seen in the embryo. We don't see very much of embryonic connective tissue in us. In some areas of the body it may be seen as remnant, like for example the pulp of your tooth has a bit of embryonic connective tissue left behind in it. When the child is born, the umbilical cord has embryonic connective tissue, but otherwise it kind of disappears more or less after we are born. Then we have connective tissue which may be loosely arranged, and usually when something is loosely arranged it kind of tends to, this kind of connective tissue tends to act as packing material because it's loose, so you can kind of push it in between spaces. And then we have connective tissue which is more compact. So when it's more compact, instead of packing material, this is used in areas where you need to withstand stress. So you want that area to be firm and compact. So that's dense connective tissue. And then we also include cartilage, bone, and blood in connective tissue. So they have their they are separate. So they don't come under loose or dense connective tissue. They just have their own separate nomenclature here. So let's first look at loose connective tissue. As the name suggests, loose, everything is very loose. So you have a lot of spaces in between. One example is something called loose areolar tissue or just areolar connective tissue. So you'll see all three components. You'll see the ground substance, which is this area. You see the fibers, collagen, some elastic. You see the cells, which may be fibroblasts. You may see some lymphocytes. Look at the amount of space between these fibers. So this space is all usually filled with almost liquid to very semi-solid jelly-like material. When you go shopping and when you're cleaning, for example, if you buy chicken and you clean chicken, you get some kind of a horrible sort of slimy substance which comes out. That's like loose connective tissue, okay? You obviously can't see in that, you can't see the cells and the fibers, but that's loose connective tissue. So you can understand how it kind of can form packing material. Another type of loose connective tissue is called adipose tissue, where the cells, they are packed closely together, giving what kind of appearance? Chicken wire appearance. It looks like a chicken wire. They give a chicken wire type of appearance. The cell is full inside the cell, which is called the adipose site or adipose cell. This area is full of fat. So when it's full of fat, what it does, it tends to push the nucleus to one corner of the cell, the periphery of the cell. So the nucleus is usually peripheral. In a slide, the cell looks empty because when we use alcohol to process the slide, the tissue, and then we put it on a slide, the alcohol removes the fat, so the cell looks very empty. So you would have seen this very empty looking cell, chicken wire appearance. Now, even though the cells are tightly packed with one another, they can be squished and take any shape. They can expand, they can be squished. So that's why it forms good packing material. Usually you see adipose tissue, like you see it under the skin, you see it around blood vessels, around organs. Again, helps to protect them, keep them in place, anchor them, and of course as packing material. So here is areolar tissue. So this is an actual slide. Here is adipose connective tissue. And then this is reticular connective tissue. Similar to areolar, only since it's found in lymphatic organs, you'll see a lot more reticular fibers. And we have to use a special stain to stain them. So you can see these short black threads. These are reticular fibers. And in lymphatic tissue, the predominant cell is lymphocyte. So you'll see a lot of lymphocytes in them. So otherwise it's still loose. The only difference being the type of cells and the fibers you see are different from areolar. Okay. Okay, I told you this. The function of loose connective tissue is either packing material or providing a framework. So we can skip this question. So. Okay, good. Now, compared to loose connective tissue, let's look at dense connective tissue, where it's more compact. Now, here in dense connective tissue, where the fiber bundles are more compactly arranged, they could be arranged in a regular pattern. That means all the fibers run in one particular way. Or the fibers could run in a haphazard direction. So some fibers run in a vertical direction, some in a horizontal, some in an oblique direction. So when we take a section, the fibers all get cut differently. So based on how the fibers run, we call it dense irregular connective tissue, where the fiber bundles are not running in the same direction. So here you can see some running horizontally. These guys here were probably running the other way, and so then when we cut, we sort of just see sections of them. And this is dense regular connective tissue. Notice how all the fiber bundles are running in one direction. So notice here, dense regular, all running in one direction. This is dense irregular. So it's still more compactly arranged, but look at the fiber bundles, all cut differently. Now, dense connective tissue, its function would be, because it's more compact, it's to withstand stress. So when you have fiber bundles running this way, you have some running this way, you have some running this way, that means it can withstand stress from different directions. So dense irregular is to withstand stress coming from different directions. Stress, or what are called tensile forces, from different directions. So this kind of connective tissue is seen in, for example, in where you have a joint between two bones. So let's say this is one bone and then this is another bone here. So you have a capsule which surrounds the joint, keeping the bones together and protecting the joint. So some of the fibers run in this direction, some fibers run in this direction, some may go like this. So this adds more protection. It also helps the joint withstand stress from different directions. So joint capsules, capsules of organs, around organs you can find dense irregular connective tissue. In the dermis of your skin, again those who were in the lab saw that, in the dermis of the skin you see dense irregular connective tissue. Some areas where you see that. Dense regular connective tissue is seen in tendons and ligaments. So tendons connect muscle to a bone. So imagine if this was a bone and here's a muscle, and then the tendon attaches the muscle to bone, and ligaments attach two bones to one another. So this is a ligament attaching the top to the bottom bone. So here the fibers run in the same direction. So therefore they can withstand stress in one direction only, or tensile forces in one direction. And as I said, examples are tendons and ligaments, where you see the fibers running in one direction. Then we come to cartilage. Now, cartilage is also connective tissue, but some things to remember about cartilage. It kind of is in a little different, the arrangement is a little different. It still has cells, it still has fibers, it still has ground substance, but there is something else you need to know in cartilage. One is the cells of cartilage are called chondrocytes. So those fibroblasts which will form cartilage, they actually get changed and they are known as chondrocytes, because the word chondra means cartilage. These cartilage cells tend to lie in a space which is known as lacuna. So cartilage cells tend to lie in a space called lacuna. So imagine there's a little space inside the cartilage and then the cell lies here. So here's the cell lying inside the space. The space is known as lacuna. So imagine, like I told you, you had jello and I put those M and M's in it. So if I took an M and M and you just sort of, made that M and M make a little hollow space for itself in that jello, that space that it made for itself would be called a lacuna, and the cell would lie inside that, okay? So one feature about cartilage is that the cells lie in spaces called lacuna. Lacunae is plural. Also, cartilage, wherever you see cartilage, the outer aspect of the cartilage is covered by a layer. It's like the skin of the cartilage, not your actual skin, but this layer which is covering a cartilage piece, the outer layer which kind of goes around the piece of cartilage, is known as perichondrium. Peri means around, chondra, remember, meant cartilage. So surrounding a piece of cartilage on the outside, going all around, is this layer called perichondrium. This perichondrium has actually got cells in it. They are like stem cells, which help cartilage in regeneration. So if cartilage was damaged anywhere, the perichondrium would send cells inside to regenerate that cartilage. So perichondrium is very important. Apart from being protective, like being the outermost layer, that layer also has blood vessels, so it provides blood supply to the cartilage, because blood vessels are only present here in the perichondrium. They don't travel inside. So nutrients travel by diffusion into cartilage, which is why I told you cartilage doesn't have a very good blood supply, because the inside of the cartilage doesn't have blood vessels. Blood vessels are only present in this perichondrium. So perichondrium is needed for regeneration. It also provides the blood supply to cartilage. But other than all of this, cartilage has it has ground substance, it has fibers, and it has cells. The difference being cells are called chondrocytes. Now let's look at the three types of cartilage. They are hyaline, elastic, and fibrocartilage. When you look at hyaline cartilage, there is ground substance which has organic and inorganic material in it. You know, nowadays you may have seen commercials which say chondroitin sulfate, oh, build up your cartilage, get chondroitin sulfate, hyaluronic acid. You may have seen, heard of those things. These are organic components which are needed for cartilage. And you have inorganic substances which are present, like the salts of calcium, phosphorus, and so on. So this area here is the ground substance. In hyaline cartilage, you also have fibers which are collagen fibers. So you'll find collagen fibers, you should find collagen fibers going across. But what happens is the collagen fibers and the ground substance, when you look at that cartilage under the microscope, the refractive index of that ground substance and collagen fibers is the same. So we cannot make out the fibers separately from the ground substance. They both look as one. So it gives a very glassy, homogeneous appearance. So that's what I mean by homogeneous. That means the fibers are not seen separate from ground substance. The reason being their refractive index is the same. So when you look at it under the microscope, because light gets refracted, we can't tell the difference. Then the cells, these chondrocytes in hyaline cartilage, instead of lying singly in a lacuna, they tend to kind of come together, and they look like eggs in a nest. So they come together. So you may have 4 cells coming together, or you may have 3 cells, 3 chondrocytes together, or 2 like that. So since they look like eggs inside a nest, these are known as cell nests. So in hyaline cartilage, it's very typical to see cell nests lying in a lacuna. And actually around the lacuna, you will see like a little darker area. You'll see a little darker staining, like a halo around that lacuna. You should know where hyaline cartilage is present. Can you give me examples of where you see hyaline cartilage? Very good. The ends of long bones, where it is known as articular cartilage. So at the ends of long bones where they form joints, it's called articular cartilage. So imagine if this is, let's say this was the lower end of the humerus. This bone here is the radius, and this bone here is the ulna. Okay, this is your elbow joint. So at the end of the radius, at this upper end of the radius here, and where it's close contact to the ulna, at this end of the ulna, and the lower end of the humerus, these will be areas where the bones will come in contact to form the elbow joint. You have articular cartilage. One important feature to remember about articular cartilage, it's hyaline, but it lacks perichondrium. All hyaline cartilage has perichondrium, but articular is one exception. It lacks perichondrium. So with the result, it does not regenerate. Regeneration is very poor. Other examples of where you would find hyaline cartilage? In all of the fetal skeleton, most of the fetal skeleton is hyaline. Where the ribs attach to the sternum, that costal cartilage is hyaline in nature. In your respiratory tracts, like the trachea, larynx, that's hyaline. Your nose, the nasal cartilage is hyaline. So you have all these examples in your notes, so take a look at that. Elastic cartilage, very similar to hyaline. Cells are present as nests. But here the refractive index of the fibers, which are predominantly elastic fibers, that's why it's called elastic cartilage. So they're not collagen fibers mainly like in hyaline. You have elastic fibers here, a lot of them. They don't have the same refractive index. So you can see the fibers separate from ground substance. And these fibers kind of are running perpendicular. So if this is the length of the cartilage, the fibers kind of cut across like that. So you can definitely make out these fibers. And otherwise it's very similar, has perichondrium, you see cell nests, very, very similar. You see elastic cartilage in epiglottis, which is part of the laryngeal cartilage, and in the external ear. This part of the ear, which is called the pinna, that's where you see elastic. So you can remember E and E is for elastic cartilage. The third type of cartilage, fibrocartilage. Look at the word fibro, meaning more fibers. So there are more fibers and fewer cells. So cells are not many, usually not seen as cell nests. In fact, the cells are single and get caught between fibers. And this kind of cartilage does not have a perichondrium. So fibrocartilage also does not have a perichondrium. Hence, regeneration is very poor in fibrocartilage. It is seen in the intervertebral discs between your vertebrae. The intervertebral discs between your vertebrae. And you may have all heard of this meniscus. Have you heard of meniscus in your knee joint? You know, people always have meniscal tears. So the meniscus in the knee joint also is fibrocartilage. Regeneration very poor because no perichondrium. So let's look at a histological picture. So here look at hyaline, look at this glassy appearance. You can see these chondrocytes lying in their lacuna. Look at the staining around. But you can't see the fibers separately. Here's elastic cartilage. This part here is the perichondrium, which you're seeing. Hyaline is taken from the middle. See the fibers running perpendicular, so you can see them. And then you can see these cell nests. And here's fibrocartilage. Very few cells, lot more collagen fibers. Okay. Next is bone. Now, if you know cartilage, always try to kind of use the same knowledge. Cartilage is called chondra. Bone is called either the word osseo or osteo. Okay? So, anything to do with bone, we will use the word osseo, osseous or osteo. So, cartilage cells were called chondrocytes. So, bone cells would be called osteocytes. Okay, we have different types of bone cells, but they would, let's take one to compare with chondrocyte. They would be called osteocytes. You had perichondrium in cartilage. Here you have periosteum in bone. So just like cartilage, the outer layer was perichondrium. In bone, it's called periosteum. Periosteum is just as important as perichondrium because it has those primitive cells inside which help to regenerate. Bone is of two types, like cartilage was elastic, hyaline and fibro. Bone is of two types. Either it is very, very compact or it is loosely arranged. So we have compact bone and a second type of bone which is called spongy bone or also known as cancellous bone. So let's look at what bone. It has ground substance. Again, this has lots of inorganic and organic material, collagen, hydroxyapatite, all that is the organic material. The inorganic are the calcium, phosphorus, all those salts. In compact bone, the reason it's called compact is because everything is arranged in a compact, very orderly fashion. So what bone does is the bone cells and the primitive bone cells which actually lay down the fibers and the ground substance, those cells are called osteoblasts. So osteoblasts lay down bone, the bone matrix which is fibers and ground substance. So when they lay down bone in circles, in concentric circles which are known, and each concentric circle is called a lamellae. A circle or a plate, a concentric plate of bone is known as a lamella. So it kind of, these osteoblasts will lay down imagine one sort of plate of bone in a circular fashion, and it lays it around a central canal over here which contains blood vessels and nerves and lymphatics. So as bone is developing, there is a blood vessel which is in bone, and that actually brings these osteoblasts, which then begin to lay down concentric rings of bone plates, which are known as lamellae. So this is the first ring, then there'll be another ring going around it like this, a third ring. So as these osteoblasts keep laying down bone, some of them get caught between plates of bone and they turn into osteocytes. So osteoblasts lay down bone and they get caught, they turn into osteocytes. And the function of osteocytes is to monitor the environment. Is everything going okay? You know, is bone modeling, remodeling, deposition, is that going okay or not? So around this central canal, you can see many of these bone plates laid around. So one set of these lamellae around a central canal is known as a Haversian system. So one set, so this whole set like we see, So there are 1, 2, 3, 4, about 5 lamellae around this central canal. So this is called a Haversian system, or also known as an osteon. So in any piece of bone, so if I take, for example, let me take the humerus, and in the diaphysis, this area of the humerus, if I take a piece like this, I will see, if I take a section, I will see many Haversian canals like this, Haversian systems or osteons, okay, microscopically if I see that. It's called a Haversian system because these plates are laid around the central canal, which is also called a Haversian canal. Now, as these cells get stuck inside, sometimes in some areas more of a bone plate may be laid, and you need to remove that bone. So we have special cells called osteoclasts which remove bone. So osteoblasts lay down bone, osteoclasts remove any excess bone that is there, so they help with remodeling. And osteocytes, as I said, they monitor the environment. Now here in the central canal was the blood vessel which was present, and that's from where it's going to get its nutrition. Like a chondrocyte, even this osteocyte lies in a lacuna. So this is the space in which it lies, and here's this osteocyte. Now these cells which are furthest away from the central canal, they are going to find it very difficult to get nutrition. So in order to help them get nutrition, you have these tiny canals which connect neighboring cells and connect the immediate cells to the central canal. These little canals are known as canaliculi. So canaliculi connect the neighboring osteocytes to one another so that they can take their nutrition from the ones closest, will take their nutrition, pass it to the next one, which will pass it to surrounding cells. So here you can see these canaliculi which help to connect osteocytes to one another and spread the nutrition. So when you look under the microscope, it looks like the ring, you know, those tree rings, and these little things kind of like little threads like a spider web. So that's compact bone. Yes. Okay. That's compact bone, and it is seen in what is called the diaphysis, or the shaft of the long bone. Seen the shaft of a bone. The diaphysis, or the shaft, not the ends, but the middle part of a bone. So if you see any long bone, if I draw a bone like this part is called the diaphysis. These ends of the bones are what are known as epiphysis. In the epiphysis, we see spongy or cancellous bone. So this is what cancellous bone looks like. Look at the difference between this and this. Here you saw this orderly arrangement of these bone plates called lamellae. In cancellous bone, it's also known as trabecular bone, because instead of having bone plates like here, they are actually, these bone plates are sort of deposited in a haphazard fashion. And anything which kind of goes out in a haphazard fashion forms like a network. So the plates, instead of like I showed you being like this, the plates are deposited this way, one plate this way, another one like this, another one like this. So can you see it's like a little network? That network is, another word for network is trabeculum. So that's why this bone is called trabecular bone. And these are not called lamellae, they are known as trabeculae. But inside this you have the same thing. You have osteocytes present within lacunae, you have canaliculi, the outer part has the periosteum which is present. And because there are so many spaces inside here, these spaces are filled with bone marrow, red bone marrow. So cancellous bone is a site of bone marrow production throughout life, because it's always present in spongy bone. So it's a site for bone marrow production. So here you can see all the marrow cells, red blood cells, white blood cells, platelets formed. That's why we say that the ends of long bones are bone marrow producing areas throughout life. So next time you chew on a chicken bone, you know, the top part of the chicken, when you break into it, can you see, you know, the marrow comes out, you can see how it's easy to break into. If you look at the central part of a chicken leg, which is called the diaphysis, you know, when you break into it, it's only in the center is the marrow cavity. in a chicken you might find bone marrow in it, but quite often it might be replaced by fat, because as the chicken gets older or the person gets older, that bone marrow cavity, which is in the diaphysis, gets replaced by fat. But the ends have that trabecular bone, which is, you can crack very easily and has bone marrow throughout life. Okay? So let's stop here.